Thanks for the introduction. Hello, everyone. Thank you for joining. I'm the Sales and Application Specialist at Genomics, and I'm really looking forward to discussing how we can use and optimize 3D cell culture to elevate biomedical research. I'd like to start by briefly running through the agenda for today's talk. We'll start with a quick introduction to 3D cell culture, what it is, and what are some of the key advantages compared to 2D cell culture assays and traditional animal models. Next, we'll get into the unique tunability of LunaGel and discuss how researchers are able to precisely adjust ECM stiffness to replicate both healthy and diseased tissue microenvironments with greater physiological relevance. We'll then consider a handful of comparisons to Matrigel, including angiogenesis assays, the study of healthy versus diseased breast cancer spheroids, and liver tissue modeling. Finally, we'll get hands-on and demonstrate the platform in action before reflecting on the session and answering any questions from the audience. Let's begin by recapping what 3D cell culture is and discussing why it is becoming an essential tool in biomedical research. 3D cell culture is an advanced technique in cell biology which allows cells to grow in a more realistic microenvironment, complete with cell-cell and cell matrix interactions. Unlike 2D culture, where cells grow in a monolayer with limited spatial context, 3D culture enables cells to grow in three-dimensional structures, providing a more physiologically relevant representation of how cells behave in vivo. As we all know, the historic workflow for disease modeling and preclinical drug discovery has been 2D cell culture assays followed by animal models. Unfortunately, the results for this approach speak for themselves. Simufilm was a recent experimental drug targeting Alzheimer's disease. While in vitro studies using 2D cultures and mice models suggested that simufilm could restore the normal shape of filamin A protein, potentially improving neuronal functions, simufilm failed to show clinical benefit in phase 3 clinical trials, leading to its discontinuation of its development in November 2024. This case, like so many before it, underscores the limitations of 2D cell culture and animal models in replicating the complex environment of human tissues. I'd like to quickly touch on the FDA Modernization Act 2.0. Passed in December 2022, the Act permits the use of alternatives to animal testing for preclinical drug development, such as advanced cell-based assays and computer models. This shift allows us to use 3D models to bridge the gap between in vitro studies and human biology while reducing costs, aligning with FDA's updated guidelines, and overcoming ethical concerns tied to animal testing. With that in mind, let's take a closer look at genomic solutions and examine why scientists are stepping away from Matrigel and into a new era of 3D cell culture with LunaGel. For those of you who already have experience in 3D cell culture, you will be familiar with Matrigel. This is a naturally derived extracellular matrix sourced from Engelbreth home swarm mouse sarcoma cells. While Matrigel has historically been the gold standard for 3D cell culture applications, it presents several challenges due to batch-to-batch variability, undefined composition, and lack of mechanical tunability. These limitations hinder its ability to create reproducible and physiologically relevant 3D models, especially for advanced applications such as cancer research and drug development. In contrast, LunaGel offers a semi-synthetic alternative which is manufactured from pharmaceutical-grade raw materials to provide consistent results. Whereas Matrigel relies on thermal gelatination, LunaGel is chemically modified for photopolymerization in the presence of visible light photoinitiators and 405 nanometer light exposure. The stiffness of the resulting hydrogel is precisely tunable by modulating the duration of light exposure, which allows scientists to generate matrices ranging from 1 to 40 kPa and model both healthy and fibrotic or tumor-like environments. Let's take a closer look at some side-by-side application studies of Matrigel and LunaGel. Our first case study of today aimed to evaluate the growth of MCF7 cells, which is a well-established human breast cancer cell line frequently used to investigate tumor growth, invasion, and resistance to chemotherapy. Confocal imaging revealed that LunaGel consistently supported the formation of well-defined spheroids, whereas Matrigel resulted in irregular and inconsistent morphologies. This inconsistency may be due to the inherent batch-to-batch variability, which is a known limitation of biologically derived ECMs. Over on the right, downstream assays demonstrated that MCF7 spheroids cultured in LunaGel exhibited significantly higher metabolic activity by day 7 compared to their counterparts in Matrigel. Additional DNA content analysis also indicated elevated proliferation rates in LunaGel, suggesting a much better supporting environment for cell growth and viability. While we're on the topic of breast cancer, I wanted to quickly add to the point I made earlier about LunaGel being photopolymerizable. This unique ability allows us to facilitate the investigation of stiffness-related disease processes such as metastasis, fibrosis, and stem cell mechanobiology. Here we have a metastatic breast cancer cell line cultured for 7 days in LunaGel matrices of varying stiffnesses. In soft, healthy like ECM, you can see cells retained an epithelial morphology and exhibited low invasiveness. In contrast, in stiffer, tumor-mimicking matrices, cells underwent epithelial to mesenchymal transition and displayed enhanced invasiveness and migratory behavior. By simply cross-linking for a few minutes longer, LunaGel enables us to mimic tumor-specific microenvironments which directly influence cell fate decisions such as proliferation, migration, differentiation, and mechanotransduction. Moving on to a different area of biomedical research, let's take a look at our second LunaGel versus Matrigel evaluation. Capillary light networks were established using co-cultures of human umbilical vein endothelial cells and mesenchymal stem cells. This 3D model serves for a variety of applications, allowing us to investigate vascular development, study endothelial mesenchymal interactions, and conduct angiogenesis-related drug screening and preclinical evaluation. Confocal imaging on days 3, 5, and 7 showed superior capillary network formation in LunaGel, with early CD31 staining confirming endothelial cell presence and actin staining highlighting cytoskeletal organization and network branching. Finally, DAPI nuclear staining validated organized cell distribution within the matrix. By day 7, we can clearly see that LunaGel supported well-branched, stable vascular networks, while Matrigel resulted in sparse, disorganized capillaries with inconsistent branching, indicating suboptimal endothelial support. By enabling the formation of consistent, physiologically relevant vascular networks, LunaGel provides a robust platform for studying angiogenesis, enabling the predictive accuracy of drug response studies and advancing translational biomedical research. Let's move on to our final case study of the session, which highlights the use of LunaGel in modeling liver tissue and exploring how ECM stiffness influences cell behavior and gene regulation. C3a liver cells were cultured in 2D Matrigel and LunaGel at 0.5, 2, and 4 kPa to compare the effects of spheroid formation and iron regulator gene expression, a relevant marker in liver disease, cancer, and iron metabolism disorders. Cultures were maintained in complete growth medium for 6 days, with media refreshed every 2 days. As shown in Figure 4a, 2D cultures produced flattened, spread-out cells, while Matrigel supported loosely aggregated, irregular spheroids. In contrast, LunaGel consistently supported the formation of well-defined spherical aggregates with increasing stiffness leading to greater structural uniformity. Gene expression analysis further revealed that LunaGel promoted stiffness-dependent regulation of iron homeostasis genes. Expression of TRF1, 2, and MT2 was significantly higher in LunaGel than in 2D or Matrigel conditions, with the highest expression observed at 4 kPa, indicating a clear link between ECM stiffness and functional gene activity in liver cells. Before we wrap up today's talk and answer questions from the audience, I wanted to briefly demonstrate how quickly and easily 3D models can be established using the LunaGel platform. First, the LunaGel ECM solution is brought to 37 degrees Celsius to ensure a homogeneous matrix environment for the cells. The photoinitiator, LAP, is then reconstituted in PBS. Cells are harvested, counted, and resuspended directly in the LunaGel solution, followed by the addition of our photoinitiator, LAP. The cell-laden hydrogel mix is then dispensed into the desired microwell plate, typically 24 or 96 well. The plate is then placed within the Luna crosslinker where photopolymerization with 405 nanometer visible light takes place. Now this is where I can directly control the ECM stiffness. For example, if I want to study healthy breast tissue, I would cross-link for a much shorter period. Equally, I could set up diseased models side by side by cross-linking for an extended period with a stiffer ECM. After cross-linking is complete, we simply add culture media and any growth factors required to the plate before transferring the plate to a standard incubator at 37 degrees Celsius and 5% CO2. Within just a few minutes, we've established a 3D cell culture system which supports physiologically relevant cell behavior and long-term model development. That brings us to the end of today's session. Before we transition into the live Q&A, I'd like to briefly recap the content we have covered. We began by exploring the limitations of conventional 2D cell culture and animal models, and the growing need for physiologically relevant 3D in vitro systems, especially in light of regulatory shifts such as the FDA Modernization Act 2.0. We then introduced LunaGel, a tunable 3D cell culture platform that allows researchers to precisely control ECM stiffness using visible light cross-linking. This enables the modeling of both healthy and diseased tissue microenvironments with enhanced reproducibility and biological relevance. Through a series of comparative case studies, we demonstrated how LunaGel outperforms Matrigel across several applications, including breast cancer spheroid models, angiogenesis assays, and liver tissue modeling. Finally, we walked through a demonstration of the LunaGel workflow, showing how scientists can establish tunable 3D models in a matter of minutes, ready for downstream analysis and reproducible models. Thank you for your time and attention today. I now welcome any questions from the audience.